All right. Okay, good morning, everybody. So what we're going to do today is just, put some of the slides that I saw, maybe kind of, a little bit complicated, confusion, something like this. So I just wanted to go through this slide again. Of course, maybe you have, your own questions, but you can just feel free to ask me questions, right? And again, I think you guys would like to focus on the class notes that we talk about in the class, and also the study guide, and make sure that you understand everything that we talk about in class, right? So class notes is most important, and then a study guide. And if you feel like, you still, some part is still not very clear, and then you can, read textbook, then make sure you understand, completely understand, right? And since this multiple choice, type of questions, so I think understanding is more important than just memorizing everything, right? Is that okay that I'm going to do this kind of format for review session? Okay. All right. So the first unit that we discussed, the carbohydrate metabolism. Again, the majority of the questions on carbohydrate metabolism, maybe 4 and I can't remember 3 or 4 questions. On the absorption, digestion absorption part, but I think it would be very easy, right? You wouldn't worry too much on that part. The first I think something that we discussed, right? The 5 different metabolic pathways of glucose metabolism, and this. The 2 tables that I give to you, it's important to make sure you understand each pathways, which physiological conditions, basically fasting conditions, and is this pathways activate or inactivate or whatever, increase or decrease or something like this, and which tissues does this particular, pathways, right? Like a glycolysis, basically every cell, every tissue, every type of cell is able to do the glycolysis, right? And gluconeogenesis, only some part of the tissue, right? Like liver, kidney, right? And not the other tissue to be able to do gluconeogenesis. And glycogen basically is stored in the 2 main tissues, right? Liver and muscle, right? And also the other question that you probably already know that, why liver glycogen can be break down and release glucose into circulation but not muscle, because of lacking of the key enzyme, right? They catalyze the last steps of the glycogen breakdown, like glucose 6-phosphatase is not present in the muscle. But this enzyme is also present in the kidney, so right, the kidney does gluconeogenesis also be able to release the glucose into the circulation. So the last step, or the glucose 6-phosphatase catalyzes the reaction which converts the glucose 6-phosphatase phosphate to glucose, and this reaction is common for both gluconeogenesis and the glycogen breakdown, right? And also you wanted to know what is the function of each pathway, why we our body does this, gluconeogenesis or glycogen breakdown or something like this, right? And the rate limiting reactions in each pathway that you need to remember, and key enzymes catalyze these reactions, and how these key enzymes are regulated by hormones and substrates, right? And glucose homeostasis, and we talk about in both fat and fasting condition. So under fat condition, our body need to do is to reduce the blood glucose level, right? When we eat a meal and our blood glucose levels increase, so it's very important to maintain our blood glucose level under normal range. So under fat condition, our body deal with the high blood glucose level. We wanted to reduce blood glucose level, and so which metabolic pathway of glucose can be activated or inactivated. The purpose of this is to reduce the blood glucose level after meal to maintain the blood glucose level in normal range, right? So like a glycogen metabolism, so glycogen synthesis pathway should be increased in the fat condition, right? So we can uptake blood glucose level and synthesize and the glycogen and store in liver and also muscle. And glycogen breakdown pathway or gluconeogenic pathway should be decreased, right? So it be inhibited because we don't want to have extra glucose. We have already enough glucose, high glucose from the diet, so we need to deal with the high blood glucose level. And you just in general, you understand this whole process, and it will be pretty easier to understand which pathway need to be upregulated, which pathway need to be downregulated, right? So, and also, extend a little bit further, if we're thinking this pathway need to be downregulated and the key enzyme in this pathway should be decreased, right? Or something like this. So just, connect all this information together to make a big picture, so that will help you understand. And also we talk about the glucose transporters, right? So in each tissues, then your glucose transporter, a different type of glucose transporter is expressed at different level and responsible for glucose transport in different tissues, right? So two main glucose transporter like GLUT2 is expressed in liver and beta cell, and basically responsible for high blood glucose level, right? So when high glucose levels increase after meal, then GLUT2 is activated, so it can transport the glucose into the liver and also pancreas, the beta cell, and the consequences is that particularly in the beta cell, when we have a high glucose and be transported by GLUT2 into the beta cell and then trigger the insulin secretion. So and the GLUT4 basically is insulin dependent, right? And so you also wanted to know the tier, first tier reaction and a response to blood glucose levels after meal, and second tier response, after meal. So first tier response, basically high blood glucose level triggers insulin release, right? And that involves mainly the beta cell or pancreas, but also involved in the liver, because GLUT2 is also high in liver, right? So the transporter, but just a different purpose. In liver, transporter glucose, GLUT2 transporter glucose into the liver for the synthesis of glycogen, right? And pancreas basically stimulates insulin release. So this is the first tier reaction response, and then the second tier response is that the insulin, right? Once insulin levels increase, and insulin can stimulate GLUT4 translocation and transport glucose into muscle and adipose tissue, right? And this slide is very important. And then the next slide is under the fasting condition, right? We don't have food available, and then we need to use the stored energy. And so in the carbohydrate metabolism that we already talked is glycogen, right? So glycogen is a stored energy, but the other important stored energy is fat, right? Triglyceride, but we have not talked about it yet. But in the carbohydrate metabolism part, the glycogen is an important stored energy. So glycogen is breakdown primarily from the liver and to release the glucose into circulation. So the primary thing that our body deal with under fast condition is to raise blood glucose level, right? So not allow the blood glucose level drop. So the liver is glycogen's breakdown that involve the homeostasis, but muscle is not involved this part because of muscle usually doesn't release the glucose, right? And also the kidney involved and together with the liver to make glucose through gluconeogenesis. So these two very important pathways that contribute to the circulating glucose, right, to raise the blood glucose level. Glycogen breakdown from the liver and gluconeogenesis from liver and kidney. And glucose transporters that you probably want to know, different transporter, how they are distributed in different tissues and what they are function and how they are regulated, basically how they regulate by glucose level or by insulin, right? Depend on the transporters. So I'm not going to go detail there. And also this table, I think is very, very important. Different transporter and tissue distribution and their function. Just wanted to remind you that in the small intestine, right, SGLT1, right, is the transporter that, and also group 5 is the transporter, right, that transport glucose and also fructose. And group 2 is also present in the, if you still remember the small intestine, right, on the other side of the enterocyte, the transporter inside of sugars across the basolateral membrane and to the portal vein. And this is just how group 2 regulates the glucose metabolism or blood glucose level in the fat condition, and I described, right, involve the two tissues, liver and pancreas. And liver is basically promote the glycogen store, and in the fat condition, right? And beta cell is trigger or stimulate insulin release. And this is also important slide. I think you wanted to remember the how insulin is stimulated by glucose. The sequential event you want to know, right? First of all, glucose uptake by group 2, and then glucose then goes to the glycolytic pathway and produce ATP, so causing ATP to ADP ratio increase and causing the membrane depolarization and calcium channel, the potassium channel actually open and causing the efflux, right? So potassium go out of cells and calcium and go inside of cells and causing increased calcium concentration. This eventually stimulating movement of insulin granule, secretory granule, and increase insulin secretion, right? So, and the first reaction I just also described already that involved the group 2, right? And two tissue, basically one tissue actually here, as beta cell, right? And pancreas and causing insulin secretion. And secondary response involved the muscle and adipose tissue, and group 4 stimulate glucose uptake into this tissue. Okay, and we also talk about the GLUT4 translocation, how insulin regulates GLUT4 activation, basically GLUT4 transport, right? Translocation from inside the cell to the cell surface and can transport glucose. Fat fasting conditions, and in both adipose tissue and scalp muscle. But under the muscle, in addition to the insulin regulation, GLUT4, we also mentioned the exercise like muscle contraction can also increase the GLUT4 translocation to the cell surface, so that allow the glucose being transported into the muscle cells. And this regulation is kind of independent of insulin, right? Even you don't have a high level of insulin, the muscle contraction itself can promote GLUT4 translocation. And the liver metabolism, and we also talk about the, each tissues, they have their own, metabolic characteristics. That means each tissue, it does different glucose metabolism. So liver basically is most comprehensive tissue, can does almost 5 different metabolic pathway activity of glucose. I can do the glycolysis, gluconeogenesis, glycogen breakdown, glycogen synthesis, and pentophosphate pathways, right? And you also need to know, that like metabolic metabolite flow, for example, the liver basically like release by making glucose under the fasting condition, making glucose and release glucose into circulation, and also the take up of lactate from the circulation, which initially from red blood cells and muscle, right, to make glucose under the fasting condition. So again, depend on the physiological condition, right? The fat condition, liver basically uptake glucose from the circulation, right, and make glycogen. So this kind of, things you also wanted to understand. And the muscle cells basically can do the glycolysis and the glycogen synthesis and glycogen breakdown pathway, right? And not much like gluconeogenesis activity in the muscle. So muscle basically does not do much gluconeogenesis. And brain basically can usually complete oxidize the glucose, right? So, and brain cell also heavily reliant on glucose for energy, and uptake of glucose and completely oxidize to CO2 and water, and through all the way down the TCA cycles. But brain cells usually don't do the glycogen synthesis or glycogen. Don't have much glycogen stored in the brain cells, right? And will also not do the gluconeogenesis. So you also wanted to be able to recognize, the diagram like this, whether this is based on the metabolic pathway, right? Whether this cell is brain cells or muscle cell, liver cell, whatever. And red blood cells, and again, because lacking of mitochondria, so only does the glycolysis for energy, and the product of a glycolysis is lactate, and lactate basically is released into circulation, right? And fat cell is kind of a little bit different from the other type of cells, and under fat condition, fat cell metabolize glucose through the glycolytic pathway, basically for synthesis of glycerol backbone for triglyceride synthesis. But under the fasting condition, the fat cell basically break down triglyceride, right, and release fatty acids, but this we'll talk about more in the lipid metabolism section. Okay, so I'm not going to go through this and these glycolytic pathways, and what you need to know is the key reactions, right? 3 key reactions and key enzymes and catalyze key reactions, and how many like ATPs or what is the product, right, of the glycolysis depend on the condition, whether under the aerobic or anaerobic condition, right? And also depend on type of cells. For example, I mean, in the quiz you already done this, right? If you compare brain cell versus the red blood cells, and the product will be different, right? And depend on again. In the ATP's production, you want to be able to calculate it, and the purpose again is just help you to be familiar with the metabolic pathway of the glycolysis, and also NADH can be generated during the glycolysis, not just the ATP, right? And NADH, but NADH production is also depend on the whether the aerobic and anaerobic condition. And because of this, this cycling, right, NADH in the reaction of the pyruvate conversion to the lactate. So again, if under the anaerobic condition, there will no NADH produced during the glycolysis. Now, just the calculation, right? The ATP production under under the aerobic or anaerobic condition, and I'm not going to go through this, and you just calculate on your own. And the and I will also mention the fructose metabolism, right? Fructose basically metabolized through a slightly different pathways, like bypass the 2 key reactions in the glycolysis of glucose, right? So that's the reason, why when we eat a lot of fructose and causing the problem, right? You have a lot of, the fatty acids being synthesized or lipids synthesized because of, they bypass the 2 key reaction that can generate a lot of glycerol 3 phosphate product, right? And which is the important product for the triglyceride synthesis. And this is also key reaction in under the after the pyruvate enter the mitochondria. So pyruvate being converted to acetyl-CoA, and this reaction, the product as the acetyl-CoA, and also generate 1 molecule of NADH, and this product can be serves as sort of negative feedback signals. When you have a high levels of acetyl-CoA or NADH is generated, and these 2 substrates can go back to inhibit the pyruvate dehydrogenase. This sort of a regulative mechanism to allow not and produce too much the energy, right? And acetyl-CoA is again the key molecule that can be generated from oxidation of 3 macronutrients, right? Glucose, amino acids, and fatty acids can all be metabolized to generate acetyl-CoA, and acetyl-CoA can have a different metabolic fate. Can either enter the TC cycle for complete oxidation. So acetyl-CoA is the molecule that enters the TCA cycle, right? Not the pyruvate. And acetyl-CoA can also be used as the blocking, the starting molecule for synthesis of other metabolic compounds like fatty acids, ketone bodies, and cholesterol can be all synthesized from acetyl-CoA, right, which we will talk about a lot in the lipid metabolism. And this is a TCA cycle, and you come together with the glycolysis and oxidative phosphorylation, or they put all information together. So this is a very nice a diagram, right, to see how glucose and the other nutrients being completely oxidized and produce ATPs. So and also I wanted to emphasize or mention again the nutrient metabolism, like glucose is metabolized to produce ATPs, right? And ATP can be produced immediately through the substrate level phosphorylation. We know that, right, in the glycolytic pathway, but also in the TCA cycle. But most importantly, this nutrient metabolite can produce the NADH and FADH reducing equivalence, right? A lot of it reducing equivalence is produced, particularly during the TCA cycle. And this reducing equivalence can be further, what I try to say, convert to the ATP in the electron transport chain, right? And it's not like it immediately produce ATP after finish the TCA cycle, but okay. So the TCA cycles, and what you need to know is the ATP production, reducing equivalent production, so then you will be able to calculate the ATP numbers. And most importantly, I think the TCA cycle, which molecules enter the TCA cycle. So the acetyl-CoA, right, is the molecule that enter TCA cycles. And also the first reaction of the TCA cycle is the acetyl-CoA together with the oxalic acetate condensed to citrate, right? So this is the first reaction in the TCA cycles, and TCA cycles also connect the metabolism of the three nutrients like glucose and fatty acids and amino acids, right? And you know that citrate is I highlighted several key molecules of intermediates, right? In the TCA cycle, you try to remember this, how this intermediates connect with the other metabolic pathways. So like citrate basically is key intermediate in TCA cycles can be used for fatty acid synthesis or go to the glycogenesis pathway. And oxalic acetate is a key molecule that can leave the TCA cycle and for gluconeogenesis, right? The alpha-ketoglutarate, the succinate, and this intermediate basically is generated from amino acid metabolism and join the TCA cycles. And this TCA cycles, it goes through part of TCA cycle for gluconeogenesis. So if you still remember that the gluconeogenesis, the TCA cycle not only oxidize all the nutrients, but also involve the gluconeogenesis, right? Particularly from the amino acids as the precursors. So TCA cycle, part of TCA cycle is also part of a gluconeogenic pathway, right? From the amino acids. So this kind of information you also need to know. And also we talk about catapolysis and anapolysis, right? Remember. So anapolysis basically is the the pathway that can add the intermediate to the TC cycles, and catapolysis, for example, like this pathways, right? Glutamine can be metabolized to ketoglutarate to add in the TC cycle. So this pathway usually we consider as the anapolysis, right? And the pathway, for example, citrate or oxaloacetate basically leaves the TC cycle, right? And for the other pathway of metabolism. So this pathway usually is called a catapolysis, right? Catapolysis pathway. And ATP production, I'm not going to go through this. Oh, okay, so this is the slide from the first unit, and let's do the second. Any question? Okay, so now we second the gluconeogenesis, right? So TC, respiratory chain or electron transport chain, also the phosphorylation, so several different names, but all talk about same thing. So you know that four complex, right, in the electron transport chain. So electron transport chain basically located in the inner mitochondrial membrane, right? The main function of electron transport chain is to convert energy from NADH or FADH, reducing equivalent, to ADP, transfer energy from NADH or FADH to ADP and to produce ATP, right? And complex 1, 3, and 4 are the pump, proton pump, right? And 2 is not, because it's not localized, on the membrane, mitochondrial membrane. And this 3 complex basically can pump a proton out of a inner mitochondrial membrane, so generating the proton gradient. And this proton gradient contains energy, so each gradient has enough energy for 1 ATP production, right? So and NADH basically transfer the electron to the complex 1 and then 2 and 4. So then one NADH molecule basically be able to produce 3 ATPs, right? And FADH bypasses complex 1 and only goes through the complex 3 and 4, so only produces 2 ATPs, right? This is kind of just general concept about it. And gluconeogenesis, you need to know what it are the precursors and what is the purpose of gluconeogenesis, is to produce glucose, right, from non-sugar precursors, and occurs primarily in liver and kidney and during the fasting, and can involve both mitochondria and cytosol. Still remember, the gluconeogenesis. Sorry, I finished this precursors, right? And the bypass reactions. Again, gluconeogenesis is not exactly opposite, direction of a metabolic flow of the gluconeogenesis, because of 3 key reactions. Irreversible reaction in the glycolysis cannot be converted by the same enzyme. So the we need a different set of enzymes to do the gluconeogenesis to overcome these three key reactions in the glycolysis, right? So basically, we have 4 enzymes that involve the reversal reaction in the gluconeogenesis, right? So you try to remember these 4 reversal reactions, not just the enzyme, but also the substrate and the products, right? And this just overview, this 4 bypass reaction gluconeogenesis. 2 occurs in the mitochondria, and the other 2 in the cytosol, and whether the gluconeogenesis will go through the all 4 reactions or whether just the 3 or 2 reactions depend on the precursors, right? If you still remember, for example, if pyruvate or lactate as the precursors, and then will involve the 4 reactions, right? 2 in mitochondria and 2 in the cytosol. And if we use glutamate as the precursors, that will involve TC cycles, but only involve the 1 reaction, key reaction in the mitochondria and 2 reactions in the cytosol. And if we use glycerol as the precursor, then will only involve the 2 reactions in the cytosol, right? You still remember me mentioned that. And this is just the enlarged diagram. And 2 cycles and Cori cycle and alanine cycle, right? You need to know where what is this basically is the gluconeogenic pathway, right? So which precursor that involve Cori cycles and which precursors involved in the alanine cycles. The Cori cycle is basically lactate, and lactate comes from muscle and red blood cells and being transported to the liver and to make a glucose, and glucose then released into circulation for the other tissue use, including red blood cells and the brain, right, during the fasting condition. And alanine, the alanine is basically released from muscle and picked up by liver to make a glucose, and then released back to the circulation for other tissue use. This is just great. Glutamate as a precursor for gluconeogenesis, as mentioned, right? Only involves 3 reactions and also TC cycles, because glutamate is converted to alpha ketoglutarate, enter the TC cycles, and goes through some of the TC reaction being converted to oxaloacetate, right, and leaves the TC cycle and join the gluconeogenic pathway. So bypass the pyruvate kinase catalytic reaction, so bypass this key reaction, right, in the mitochondria. And I think this slide I just wanted to mention. I think we talked about it already, right? How glutamate is used for gluconeogenesis, the reaction, the pathway, gluconeogenic pathway. And this clinical correlation, we talk about 2 cases, right? And you want to understand how, what kind of problem that the patient will have if they don't have PAP-CK, right? Basically hypoglycemia and have a lot of lactate and accumulate in the circulation, and also we use a lot of glycogen, right, breakdown. So again, gluconeogenesis, glycogen breakdown are the 2 key pathways that produces can make the glucose, right, and to raise the blood glucose level. So if one pathway, like a gluconeogenic pathway, is not working, and we compensate to make it harder, right, to break down the glycogen. So the more glycogen being used in this patient, the glycogen, causing glycogen depletion. And The glucose 6-phosphatase is a different issue in terms of the glycogen store. Similar to the PEP-CK deficiency, glucose 6-phosphatase deficiency can also cause the hypoglycemia and increase lactate, right, which understandable. And but the actually causing the glycogen accumulate, and the reason, if you still remember, the glucose 6-phosphate is accumulated if we don't have a glucose 6-phosphatase, right, to convert it to the glucose. So causing the glucose 6-phosphate accumulation, which is the substrate that can inhibit glycogen breakdown enzyme, like glycogen phosphorylase. So glycogen cannot be efficient breakdown, so then accumulated. So this case is different from the PEP-CK deficiency in terms of glycogen store, right? And the kidney is also important organ that also be able to does the gluconeogenesis. Basically, mostly use amino acids, right, as the precursors, and very important when we undergo the long-term starvation, right, prolonged starvation. The kidney becomes important, and liver was the first tissue, right, that break down that does the gluconeogenesis. And this anaeroprosis and ketosis, I think I described this already, right? So you just the concept, right, what process is called ketosis and which process called the anaeroprosis. And pentophosphate pathway, which is the last. Oh no, not last. So the substrate you need to know is the glucose 6-phosphate, right? So this pathway uses the glucose 6-phosphate as a substrate and converts it to the 5-sugar phosphate, 5-sugar carbon phosphate. This is the main purpose, and it's also dealing with the pathway that NADH is produced, okay. So now we're going to talk about the regulation, right? Regulation, basically 3 different levels, right? The glucose transport level, right? It can be controlled, right, by transporters. But of course, transporter is regulated by glucose level or by insulin, and then substrate cycle levels. Substrate in each metabolic pathways can also regulate enzyme activities, right? And lastly, the hormonal regulation. So hormonal regulation, and I would think is on the top, right? And then it regulates the key enzyme in each pathway, and then we're causing the change in the substrate. So substrate levels change and can also add on top of the regulation, hormone regulation and glucose transport regulation, right? And different glucose transporters that have a different affinity for glucose and Km. You probably wanted to get familiar with this. Focusing on again, GLU2 and GLU4, right? And glucose transporters, 2 and 4, particularly, and get actually connected with the different metabolic enzymes. I think I did the next couple of slides. So, I'm not going to details. And glycolysis and gluconeogenesis, these are the 2 metabolic pathway and does the opposite function, right? One is like oxidize glucose, and the other one is synthesize, right? Generate glucose. So how this two pathways are coordinated regulate, that means when under the certain physiological condition, we just want to one pathway being activated and we need to inhibit the other pathway. How this could happen, right? Basically regulated by hormones and substrate. So you need to understand how insulin regulates the key enzyme in glycolysis and gluconeogenesis. Just take the first example, like glucokinase, right? Insulin basically activates the key enzyme in the glycolysis, but inhibits enzymes in the gluconeogenesis. So this, a general idea. And hormonal regulation, want you to connect the hormone secretion to the physiological condition, right? The fasting, insulin increase and glucagon decrease, and fat condition, insulin increase and glucagon decrease, and fasting condition, insulin decrease and glucagon increase, right? And the hormone regulates the pathway in different, right? The opposite, actually, glucose versus glucagon. So again, in general, insulin should increase glycolytic pathway, right? So glycolytic key enzymes, and inhibits gluconeogenic enzymes. And so net effect of insulin will be decreased blood glucose level. And in the fasting condition, glucagon and the other hormones will be increased, and you can, and just make kind of a prediction that gluconeogenesis pathway should be upregulated, right? And so, and increase the blood glucose level. And also, I wanted you to know that the although we talk about glucose and gluconeogenesis glycolysis, but since we learned actually 5 different metabolic pathways, right? So regulates glucose not only just glycolysis and gluconeogenesis, but glycogen breakdown, glycogen synthesis pathways also regulate or work together, right, to either reduce blood glucose in the fat condition or raise the blood glucose level under the fasting condition. So you also wanted to remember that insulin or glucagon should regulate all these 4 different metabolic pathways: glycolysis, gluconeogenesis, glycogen breakdown, glycogen synthesis, right? All right, you need a break? Okay. Okay, so yeah, so just the other 2 key reactions, how enzymes are regulated, right? Like hexokinase. Of this actually details. You also need to go a little bit detailed about each specific enzyme, how they regulated by hormone substrate, right? For example, glucokinase or hexokinase. And again, glucose kinase, hexokinase, they have a different tissue distribution. Glucokinase basically in the liver and pancreas, and hexokinase in the scalp muscle. And how they work together with which type of glucose transporter, right? Like hexokinase, because it's in the muscle, so basically it's coordinate with GLUT4, right, and regulates glucose uptake and oxidation and kind of stuff, right? And glucokinase is coordinated with GLUT2, right? And the substrate, how they, group fructose 1-phosphate, fructose 6-phosphate, and glucose, right? Insulin, how this substrate and hormone insulin regulates the glucokinase. So basically, it regulates translocation, right, from nuclear to the cytosol. This kind of process. And the glucose 6-phosphatase is relatively simple. I mean, insulin, since this is a key enzyme in gluconeogenesis, you would expect that insulin basically should inhibit this enzyme. And then the second pair of the key enzyme in these two pathways and involved in the bifunctional enzyme and the key substrate, the fructose 2,6-biphosphate, right? So you need to know which bifunctional enzyme regulates the production or degradation of this key substrate. And the other important thing is that this key substrate is not involved in any metabolic pathway, right? It's not the intermediates of the glycolysis or gluconeogenesis, right? It's like independent, the substrate, but it does very important regulative function, right? Regulates key enzyme in both glycolysis and gluconeogenesis, but they does a different regulation, depend on the concentration. So insulin and glucagon basically regulates production or concentration of this key substrate, right? And the key substrate then, in turn, regulates, directly regulates the key enzyme in both pathways, right? So, on this slide, you want to go detailed, and this just tells us, how insulin and glucagon regulates which bifunctional enzyme activity increase or decrease, and how the key substrate is levels is regulated by insulin and glucagon. And it's just the overall picture, right? Under the fat fasting condition, the fructose 2,6-biphosphate, the concentration is actually different. For example, in the fat condition, this substrate levels increase, right? And fasted condition, substrate levels decrease. So then causing the different consequence results. And this is pyruvate key enzyme, right? And how they regulate by insulin and substrate. And PEP-CK is a little bit different from the other enzyme. They only regulate by hormones at the gene expression level, right? Not like a modification is involved. Okay, now the last part of the glucose metabolism is the glycogen metabolism. So glycogen breakdown and synthesis pathways and regulation. Yeah. Right, by gene expression, only regulating upregulated genes, right? Gene, the gene transcription levels, right? So this is important slide, right? You wanted to know, basically it covers all the pathway, right? Breakdown or synthesis, and which enzymes involved, and it just want you to remember too. The key enzyme, right, in each pathway, like glycogen synthesis, the key enzyme in the glycogen synthesis pathway, and glycogen phosphorylase as the key enzyme in the glycogen breakdown. And that glycogen synthesis also involve like UDP, right, and blue glycogenin, as primers and synthesize first 8 glucose chain, and then glycogen synthesis take over to complete the synthesis of and the branching enzyme, right, also involved to complete the synthesis of a compact glycogen compact. And also the two, actually the substrate, right, is also important. The glucose 6-phosphate and full glucose both can inhibit the glycogen phosphorylase, right? So, which also makes sense if we consider all things together, like during the fat condition, we have a lot of glucose available, and we generate a lot of glucose 6-phosphate. And once these two substrate levels increase, then we will break down the inhibit the glycogen breakdown pathway, right? That makes sense. So this is one of the mechanism, the regulation, right? And the phosphorylation, I told you that the cyclo-AMP is a very important, the intracellular, the secondary, so-called secondary signal, whatever. So cyclo-AMP level is increased during fasting or by norepinephrine stimulation. So cyclo-AMP levels increase basically inhibits glycogen breakdown. Inhibit glycogen synthesis, but promote glycogen breakdown, right? So just try to connect the cyclo-AMP to the fasting condition and to the epinephrine level. And insulin basically reduces the cyclo-AMP level, right? So we can reduce the glycogen breakdown pathway and stimulate glycogen synthesis pathway. And I guess this is the last slide. You could just make a comparison, fat fasting condition, which hormones regulates, which pathway, which enzymes upregulates, downregulates, right? So that should be easier. And then also the last slide, we make comparison, the liver and muscle, their glycogen store, and the regulators, right? They are slightly different. So, for example, hormones, glucagon only regulates liver but not muscle, right? Glycogen metabolism. And in terms of substrate, the free glucose regulates liver but not muscle, and muscle glycogen metabolism is regulated by 5-AMP, right, or something like this, And also the role of the stored glycogen in terms of the contribution to the circulation glucose is also different, right? Liver be able to release glucose from glycogen breakdown, but muscle not. And again, reason is the glucose 6-phosphatase enzyme, right? It's lacking in the muscle. Okay, so that's all I have for the reviews. And do you guys have any question? And I have actually 3 slides of the kind of true to false questions, right? Let's go through it, right? So first question, glucose is oxidized to either lactate or pyruvate via glycolysis. It's true or false? True, right? And second, oxygen is not required for glycolysis. True? Not required. I mean, glycolysis, we're just thinking about from glucose to either pyruvate or lactate. This true, right? Yes, true. And 3, and all cells can do glycolysis for producing energy from glucose. True, right? And 4, glycolysis is the first step of glucose metabolism, it breaks down glucose and generates intermediates, which can be further metabolized or converted to intermediates of other pathways. For instance, lipogenesis. Is this true? Yes. 5. So one of the limiting steps of glycolysis is the glucokinase catalyzed phosphorylation glucose to glucose 6-phosphate. True, right? It's easy. Yeah. Okay, now about the TCA cycles. So first, TCA cycles generate ATPs. Yeah. And second, TCA cycles is common to oxidation of carbohydrate, fatty acids, and proteins. Yeah. And 3, pyruvate is the starting molecules of TCA cycles. Which molecules? Yes, you're right. And number 4, TCA cycles is the only cycle pathway that produces reducing equivalent and ADH. Which other pathway can produce? No, that is NADPH. Yeah, glycolysis, right? Yes. So, 5. TCA cycle is the ending oxidative pathway for all 3 macronutrients. True. How many you think is true? Not many. So the rest of you guys think it's false, right? Why? Why is not the final? So you're thinking electron transport chain will be the final steps, right? Yes. Okay. But my answer, I don't know. Oh, I thought it's true. I can change my answer. Right? As long as you guys answer reasonable. I think yes. Okay. So I mean, in terms of chemical reactions, right? It stopped at TCA cycle, right? But the electron transport chain, involve like electron transport, right? And then produce ATP energy. So I think both could be true, depending on how we understand. So this is not a true and false. The right answer, whether it's the right answer or not, is not our final purpose. I just want you guys to understand, the whole process, right? So you guys right. It's not the final if we consider ATP production, right? It's okay. So 6. So the first reaction in TCA cycle is the condensation of oxaloacetate and acetyl-CoA forming citrate. True, yes. Okay. 2 more. Okay, which one? Oh, I will post it. That slide, right? Okay, so the last 2. So the end product of TCA cycle are CO2 and water. True, right? Okay. And oxaloacetate and citrate are the intermediates that can be removed from TCA cycle for gluconeogenesis and lipogenesis. True, right? Okay. We have a kind of question like this, but maybe slightly in terms of the difficulty, probably a little slight higher than this one. Okay, so any questions you have specifically? Anyone? No? Okay.